Hey everyone, in the last video we took a look at fertility factor or F factor mediated conjugation. In this video we are going to take a look at how an F plus bacterium, an F plus E. coli cell can become what we call an Hfr cell for high frequency of recombination. And if you're hopping into this video without watching the others, again, my hands are pretty disgusting from doing some plumbing work at my house yesterday, and I can't get this grime off of my fingers. Hopefully by the time I make the more videos next week, it will be gone. Okay, so high frequency of recombination. What does this mean? Where does this name come from? How does an F plus cell become an Hfr cell? So high frequency of recombination. Actually, it's pretty easy. So let's say this is the F factor, which is just a plasmid in F plus E. coli cells. What happens is somehow this integrates into the chromosome. And again, we will talk about molecular mechanisms of recombination in the next part of the course. But for now, all we need to know is the F factor integrates into the chromosome. And the F factor can integrate in many, many different areas in the chromosome. So let's say this one integrates right here. Now remember, we have the OOT, the origin of transfer, in the F factor. Now let's say that OOT is right here. And it's oriented in a way that when the nick is made, the DNA will be unwound in this direction, clockwise. Now, like I said, okay, so once this happens, F factor is no longer free in the cytoplasm of the bacterium. It's integrated into the chromosome of the bacterial cell. When that happens, okay, now it is an Hfr cell. And like I said, the F factor could integrate in any number of areas. And let's say it integrates in a way that, okay, we're near this gene called LAC, and this gene has the LAC plus version, the LAC plus allele there. What that means is, you know, when E. coli has a functional copy of this gene, it can use lactose as a carbon source. Let's say way over here, there's another gene, Pro plus, and this gene is required to make the amino acid proline. Now, in a previous video, we showed how the F factor can mediate recombination between an F plus strain and an F minus strain. So the F factor, when inside the chromosome, can also mediate recombination between an Hfr strain and an F minus strain. So let me diagram that down here. Please review the previous video to see the basics of how recombination works. But let's just jump up right to the stage where the cells are side by side. We have the conjugation tube. This is the Hfr cell, no longer calling it F plus, now it's Hfr. This is the F minus, so the DNA is going to go from the Hfr to the F minus strain. Here is the chromosome. I'm going to diagram it as a small circle right here so I don't take up the whole cell. So here is the F factor, and remember I said the OOT was oriented, so if it's cut, the strand is going to be unwound in the clockwise direction. And all the bacterial genes are on the bacterial chromosome. We only mentioned two, LAC plus and Pro plus. And I'm getting a call, which is weird. I never get a call here. So I'm just going to ignore that. Sorry about that. So again, so this is the bacterial chromosome with all 3,000 or so genes in E. coli, but the F factor is right here. We're only concerned with LAC plus and Pro plus for this example. Now, when the F factor mediates recombination between the HFR and the F- strain, what happens is we still get the nick at the OOT in the F factor, and then when it starts being unwound, what happens is, okay, I'll make this area double-stranded. I'll make the whole thing double-stranded for now. Is we start transferring a single strand of the whole chromosome to the F- strain. Okay, so I have the I make the nick here, proteins come in, they start unwinding and delivering this piece of DNA into the F- strain. And okay, here's the chromosome in the F- strain. And let's say the F- strain, let's call this one lac minus. It has a copy of the lac gene, but it's a mutated copy. And it has a copy of the pro gene, and it's a mutated copy. So this DNA is going to be transferred, and because the OOT is always nicked at the same spot, and the DNA is always unwound in the same way, in the clockwise direction, every time we see a recombination, a conjugation between an HFR cell with this chromosome, with this F factor integrated at this spot, with an F- cell, the lac gene is always going to be delivered over into the F- cell first, right? Because the lac plus gene is closest to the OOT, and the DNA, the single-stranded DNA, is being unwound in a clockwise direction. If the F factor was sort of integrated in the reverse orientation and we unwound this way, well then pro would be the first one to be inserted into the F- strain. But no, because of the orientation, the OOT is pointed this way. We're always unwinding in the clockwise direction, and lac plus is always going to come out, enter the cell first, the F- cell first, relative to pro plus. So eventually, pro plus will get in there too. So we'll have a long piece of DNA in here with lac plus and pro plus. Okay, and maybe the whole chromosome goes in there, or maybe this conjugation tube is broke before the whole chromosome can transfer. But as long as a piece of lac plus goes in and a piece of DNA containing pro plus goes in, these pieces of DNA can recombine, exchange places with the lac minus and the pro minus alleles. And we'll talk more about the molecular mechanism of how this happens in future lectures. But just know, as long as a copy of lac plus gets in and pro plus gets in, there's an ability of lac plus to change places with lac minus and pro plus to change places with pro minus in such a way that this F- strain can now be lac plus and pro plus. So HFR, high frequency recombination mediated conjugation, can allow this strain with functional copies of lac plus and pro plus to donate those genes to an E. coli cell with mutant versions of those same genes. So those mutant versions can be replaced with the functional copies. So in similar, I'll just touch on one more thing. The HFR in this case, of course, is the donor. F- is the recipient. And in this case, the F- is not becoming an F plus cell or an HFR cell, because it's usually not getting the fertility factor during the transfer. And even if it does, the fertility factor is not likely to insert into this chromosome right here. So unlike the F plus mediated conjugation, the F- cell is not being turned into a cell that is capable of driving conjugation with another F- cell. So that's it for this video. In the next video, I'm going to talk about how HFR-mediated conjugation was used to make the first genetic maps of genes in bacteria.